All right. So we lost UCP1 in piglets. And so, so cute. They huddle up and shiver together. And people take pictures of it and go, that's so cute. Okay? And again, they're doing that because they don't have the UCP1 pathway to generate heat without shivering, right? This is going to lead to high energy costs for pig farmers, actually. So the high piglet mortality as well, and accumulation of fat and white adipose tissue as a thermal insulator. That's one of the reasons why pigs put on that very thick layer of fat on the outer portions. You guys have all seen that probably. Yeah, you've seen pigs before? Fantastic. Okay, I can never take these things for granted, right? Yeah. The CRISPR, this has been used to insert the UCP1 gene into pig embryos. It's cheaper to raise due to the decrease in energy costs. Provide more high quality lean meat actually, because they don't build up these huge, thick fat layers. Okay. By the way, ever since I came to Tennessee, normally in the north they heat the barns because they lose huge amounts. In Tennessee they don't, and I think that's why your guys' meat down here is way fattier, like huge deposits of fat, right? Than you get up north. I think that's it. And I think that's why it's been like super fatty lately, because we had some pretty bad winters. Anyways, very fatty meat around here, right? I think that's why. So many potent and lethal poisons are going to inhibit oxidative phosphorylation in one of the following ways. The inhibition of the electron transport chain, thus preventing the generation of the proton motive force. Inhibition of ATP synthase, uncoupling electron transport from the ATP synthesis, inhibition of the ATP export. So for example, right here we have 2,4-dinitrophenol. This is DNP. This carries protons across the inner mitochondrial membrane down their concentration gradient via that right there. That's the one that would be carried. Notice it's neutral. It can go across a membrane, but it has a modulation of that can accept the protons and then release them fairly easily. The ATP is not formed because the proton motive force is continuously dissipated, and thus you would last about 40 seconds and then you would die. Okay? Xanthohumol, this is going to be a promising drug that functions as a mild uncoupler and scavenges free radicals. This may be used in the treatments of obesity and certain cancers. Interestingly enough, did you guys hear that they found an ERR general receptor that can duplicate the effects of training for a marathon? Yeah, pretty cool stuff. You can look it up sometime. If you put ER general receptor, it's SUPP something number. NADH, NADH-Q oxidoreductase. This would be blocked by rotenone and amytol right here, which is the NADH-Q oxidoreductase, generating your QH2 and thus filling your Q pool. This Q pool typically goes to Q cytochrome C oxidoreductase. This is actually blocked by antimycin A, so it cannot generate the cytochrome C, which would do the next one, right? And so then that can't go into the cytochrome C oxidase. The cytochrome C oxidase going to O2 is actually blocked by cyanide, azine N3 minus, and carbon monoxide. Anybody ever wonder one of the reasons why you die so quickly from carbon monoxide poisoning? No? Nobody wondered that? My goodness. Anyways, this is also cyanide poisoning. That's why you turn blue, is because even though you're getting oxygen, it doesn't do you any good. Okay? The mitochondrial diseases are estimated to affect from 10 to 15 per 100,000 people. As rare diseases go, those are actually pretty high. And honestly, that number is probably going to be changing hugely, because there's a lot of diseases that are starting to see efforts to reclassify them as mitochondrial diseases. For example, things like schizophrenia, is very likely a mitochondrial disease. Parkinson's, Alzheimer's, those are all probably actually mitochondrial diseases, but haven't been linked to it yet. The mitochondrial diseases are going to be primarily inherited maternally. The human eggs are going to harbor several 100,000 molecules of mitochondrial DNA, whereas the sperm only harbors a few hundred. The mutations in complex 1 are going to be the most frequent causes of mitochondrial diseases. Variations in the percentage of mitochondria with the mutation is going to lead to large variations in the nature and variety of the symptoms, as well as the time of the onset. And so this is one of the reasons why it's been so hard to actually figure out what are mitochondrial diseases, where should we target these things, is because they don't have similar onsets. You can have a variety of mitochondrial responses, and it just clouds the entire picture of where the hell this is coming from, right? When you don't have like, this develops on your 85th birthday, and it shows exactly these symptoms. Apoptosis, this is going to be a form of programmed cell death. This is going to occur in the course of development or in cases of significant cell damage. Mitochondria will act as the control centers regulating that process. Mitochondrial outer membrane permeabilization, or MOMP. I'm so tempted to call it MOPPET every time I see that. Yeah. This is going to be the process by which the outer membrane of the damaged mitochondria becomes highly permeable. It's instigated by the BCL family of proteins. Cytochrome C is the potent activator of apoptosis that interacts with the apoptotic peptidase activating factor 1, abbreviated APAF1. This is going to be leading to the formation of apoptosome. Apoptosome is going to be the protein complex that recruits and activates a proteolytic enzyme called caspase 9. Literally casp, and then ACE as in the enzyme. Okay? 9. Caspase 9 is going to be the member of the cysteine protease family that's going to activate a cascade of other caspases. Each caspase is going to, this type is going to destroy a particular target. Remember, we're causing cell death. We're given the kill order. Now, don't you find it funny that we're symbiotic? We think we're the human cells, but the ones in control are what? Fucking mitochondria. They're the ones that decide whether we eat, live, breathe, die, everything. Okay, they even affect our moods, our ability to think, our ability to feel, everything. So who's in charge? Mitochondria. And they can't even talk. Man. So the bacteria, they swim by rotating flagella that lie on their surfaces using free energy that's derived from the proton gradient. This exists across the plasma membrane. Does not require ATP, actually, which is kind of interesting because most people would think that swimming around would require the ATP, right? Guess what? By the way, kind of a cool thing is, scary, terrifying, and cool. Did you know that you actually usually act about 80% of the time? You do the actions, and then your left brain justifies why you acted. And so the reasoning you think you're doing your actions, 80% of the time usually, is not. You just did them, and then you went, I think I did them because of this. And then you go, I think I thought that before I did it. And you did not. Yeah. So surprisingly, we have a lot of our actions dictated by the right side of the brain, the mitochondria, and then the subnervous system. So MOT-A, MOT-B, these are going to be membrane proteins, each containing cytoplasmic and periplasmic domains. Approximately 11 MOT-A and MOT-B pairs form a ring around the base of the flagellum. The MS, which is the membrane and supermembrane ring, this can be a disc-like structure containing FLIG, FLIM, and FLIN proteins. This right here is the diagram of this overall thing. So right here, remember we have two membranes in these bacterium. We're going to have the outer membrane, the peptidoglycan, and then there we have the periplasm, and then we have the next membrane, and then we have the cytoplasm, okay? That's that gram negative, remember? And that sort of gets trapped because we got the two membranes. Okay, so this right here is the flagellum. This would be the filament of the flagellum. And this part's the hook of it, where it directs it. This is the L ring, P ring. The P ring actually is the peptidoglycan layer. Right here we have the MOTB, MOTA pairs. This right here is the MS ring, FLIG, FLIM is the green right here. Okay, kind of cool, huh? Ooh, flagellum. Right. So each MOTA and MOTB pair is going to be thought to form a structure that has two half channels. The MOTA and MOTB pair and FLIG are going to combine to create a proton channel. This is actually what's going to drive the rotation of the flagellum. I know you're going like, why the fuck is Dr. Clausen even talking about this right now? Guess why? Proton channel causing a motion, which translates into some sort of energy. Yeah. So again, your mechanism is going to be extremely similar to the proton-driven, rotary motion of your ATP synthase. Remember, if you go like this and you have a little whip attached to it, what motion does the whip make? A following motion, right? If you happen to get anything in the way, what happens? It gets pushed, right? And so another way of thinking about that is, what does a prop do? It just spins, right? So again, the FLIG is going to be the rotating proton carrier here. And again, that's the outer half channel, inner half channel right here. And we can see the protein uptake through the outer half channel right there, popping protons in. This is going to cause, in this case, a counterclockwise rotation of our MS ring. And this is going to then release the protons through the inner half channel. And then it's going to end up going the next rotation. And it's going to have the proton come in, driving it through the next one. Just keep doing that rotations. Now, notice that this is a bit different of a motor design than our previous one. Instead of the inner part spinning, we have the outer part spinning. Okay? So right here we have our proton gradients, the powering variety of different energy requiring processes. Again, the proton gradient is going to be a central and convertible currency of the free energy in biological systems. And here we have the proton gradient, again, correctly stated as delta P, powering different processes. For example, electron potential, heat production, NAP, pH synthesis, which is electron movements, but very specific pathways, ATP production, flagellar motion rotation, and of course, active transports. Does anybody have any questions? No? Okay. We'll leave off on, I guess that's it. How's my time? Perfect. Holy shit. Man, wow. That doesn't happen often, does it?